This is a DNA strand, and watch what happens when we zoom in. Yep, that's the CRISPR-cutting Cas9 enzyme snipping away at the double helix. It's like a pair of molecular scissors. Scissors that could help us cure genetic diseases, supercharge crops, and maybe even help fight climate change. If you want to understand how CRISPR works, stick around because today we're going to walk through exactly how scientists edit genes in the lab step by step. We made this video because we know there are a lot of misconceptions about this revolutionary technology, so we'll clear some of those up. And most importantly, we're going to have fun doing it. Let's start from the beginning. Imagine you're trying to replace one word in a really long book. You could read the entire thing from start to finish, but that would take forever. Instead, you'd probably use a find and replace tool. Well, CRISPR works kind of like a molecular find and replace tool for DNA, except instead of one word, we're replacing a gene. So first, scientists need to find their gene of interest. If they didn't, they'd be searching forever. So let's move into step 1. Design the gRNA. gRNA stands for guide RNA, and it basically acts like a GPS guiding Cas9 to the right spot in the DNA. The gRNA is programmed to recognize and bind to a specific sequence of DNA. In other words, it tells Cas9 where to cut. Using tools like benchling, scientists can analyze a genome and determine the best possible gRNA. This software predicts how well the gRNA will bind to its target sequence, which is important because if it doesn't bind well, it might cut the wrong place. That's called an off-target cut, and it's a no-go. So once scientists have the perfect gRNA sequence, they synthesize it. No worries if you don't know what that means. Just think of it like ordering something off a menu. Then it arrives in the mail, ready to go. Now that we have our GPS, let's move on to step 2. Prep the CRISPR components. Remember, we're going to use Cas9 as our pair of molecular scissors to actually make the cut. Cas9 itself doesn't have GPS. It needs directions. So scientists combine the Cas9 protein with the gRNA and voila, we have a complex that can target a specific sequence in the DNA. Think of it like putting puzzle pieces together. They fit perfectly because they were designed to. But we're not quite done yet. We still have to get this molecular puzzle into the cell. And that brings us to step 3. Deliver it into cells. There are a few ways to do this, but the main methods are lipofection and electroporation. Lipofection uses fatty bubbles called liposomes to ferry the CRISPR components into the cell. These bubbles fuse with the cell membrane and release their contents inside. Electroporation, on the other hand, uses electric pulses to create tiny holes in the cell membrane. Through either method, the Cas9 and gRNA get into the cell and bind to their target DNA. Once they're attached, Cas9 makes a cut in the DNA. Now, strictly speaking, Cas9 doesn't cut both strands. Instead, it nicks one strand. Other enzymes come in to help, and presto, we have a double-stranded break in the DNA. But how do we repair this break? That brings us to step 4, DNA repair. When cells encounter DNA breaks, they activate their built-in repair mechanisms. There are two main pathways, NHEJ and HDR. NHEJ is kind of like duct tape. It's quick and dirty and often knocks out the gene. HDR, on the other hand, is more like precision surgery. It's slower, but it allows for a more precise repair. The tricky part is that cells mostly use NHEJ to repair breaks. HDR only kicks in about 1% of the time. But we can boost HDR by providing a template for the repair process. If we give the cell a Band-Aid, it might just use that to fix the break instead of using NHEJ. The template can be any piece of DNA that has the sequence we want to insert. With a little bit of molecular biology wizardry, scientists can get their genes in there too. Now, NHEJ still happens, but if we've done everything right, we should see our edited gene as well. In the next step, we'll talk about how scientists check all this out. But first, let's look at the timeline for each of these steps. How long does it take to complete a CRISPR experiment? The answer is, it depends. A lot of factors can affect the timeline, like the type of organism, the efficiency of the experiment, and the skill of the scientist. But let's look at some examples. Bacteria are the easiest and fastest. An experiment can be completed in as little as a few days. Human cells can take about a week, and mice can take weeks. Mice are just grumpy like that. They hate being rushed. And remember, each of these steps can take several days on its own. So it's not like you can just speed things along by doing them faster. But here's a pro tip. Instead of waiting for the final step to verify the edits, scientists can use a surrogate marker. Some genes are easy to edit, so scientists can choose a gene that's easy to modify and edit that one first. If the rest of the experiment fails, they don't have to start over from scratch. They can just edit the main gene of interest instead. That brings us to step 5. Check the edits. There are two main ways to confirm edits. The first method involves traditional PCR followed by sequencing. PCR lets scientists amplify a specific region of the DNA so they can study it up close. Sequencing then reveals whether the edits were successful. This is considered the gold standard, but it takes time. Luckily, there's a quicker way to check for edits, especially for liposuction experiments. Many labs include a reporter gene along with the Cas9 and gRNA. Reporter genes have a fluorescent marker that glows green, or in this case, GFP. If the CRISPR components work properly, the reporter gene should be expressed. That means we can quickly tell whether the system is working by looking for green fluorescence under a microscope. It's like a little green flag that says, "We did it." Of course, this method only tells us whether the system is working. To actually verify the edits, we still need to use PCR and sequencing. Speaking of sequencing, that brings us to the last part of our video. Timing matters. Just like the overall experiment, each step can take a different amount of time. For example, DNA extraction and purification can take less than a day, but cell culture can take several days. And again, timing is affected by the organism. Bacteria reproduce quickly, so they're the easiest to work with. But human cells divide much slower, which means experiments take longer. Here's a rough timeline for a mouse experiment. Day 1, we inject the zygote with CRISPR components. On day 2, we culture the embryo. On day 3, we harvest the tissue and extract DNA. On day 4, we sequence the DNA and analyze the results. And on day 5, we start over. That's because every experiment is slightly different. Even if one experiment works, the next one might not. So scientists often repeat the process to make sure their results are correct. But why do scientists bother with all this work? What's so great about CRISPR? CRISPR has revolutionized gene editing, making it easier and cheaper than ever before. It's helped researchers uncover the mysteries of genetics and develop new treatments for diseases like cancer and HIV. And that's just the beginning. CRISPR could help us cure genetic diseases, supercharge crops, or even fight climate change. But with great power comes great responsibility. CRISPR raises ethical questions that we need to consider carefully. As we move forward, we need to have open conversations about the risks and benefits of this technology so that we can use it responsibly and safely. That's all for today. If you enjoyed the video and want to see a real lab demo, smash subscribe and comment DNA below for a behind-the-scenes video. Thanks for watching, and until next time, stay curious.